Hello, everyone. It is my great honor and pleasure to host Rangan Sukumar. I will have Paolo Faraboski, who is on our corporate liaison program of Santa Clara Valley section, IEEE section, to introduce Rangan. Just a couple of logistics. You are all muted because we are recording this. In the past, there was a lot of noise. And you can ask the questions. There's a button at your right, Q&A. Please type in your questions there. You can start doing it now, and Paul and I will be fielding questions later. So with all that, Paolo, could you please introduce Rangan?
Yes, well, it's my honor to introduce Rangan Sukumar, who will be the speaker for today's talk. Rangan's currently a distinguished technologist in the CT office of the High Performance Computing Group at HP. He actually came to HP through CRAY, when the company merged about a year and a half ago. And his core expertise is in data science and artificial intelligence. Prior to CRAY, he was a group leader and a research scientist at Oak Ridge National Lab, and he had the opportunity to work on some of the world's fastest supercomputers. Rangan has a PhD from the University of Tennessee. He has over 70 publications in areas of data science, AI, and various other fields. And today is going to cover a really hot topic, and basically how to fight a novel virus with supercomputing. I wonder what virus he's talking about. Rangan, the floor is yours.
Thanks, Paulo. Appreciate the introduction, and thanks, Dejan, for the opportunity to speak today. And it's my pleasure today to talk about the effort at HPE to fight this coronavirus and the pandemic. And it's an honor now to talk about the work that's been going on for the last 6 months or so. And I didn't have enough space on the slide to put in 170 different names of folks that have volunteered their time, worked extra because they knew their expertise mattered, and so forth. And several subject matter experts that I'll be referencing through this talk. Folks from the University of Alabama, Huntsville, Dr. Jerome Baudry, Kendall Byler, and APPROCH, Ryan Yates, Amachiti Boynia Iklas from University of Mississippi, and Yuri Peterson from the Medical University of South Carolina. Aaron Rose, he used to work at HPE before, and now he's at a startup, and he's part of some of the slides I'm going to talk about as well. So what I'm going to talk about today is actually work that began way before the time of the pandemic and the coronavirus. And so I wanted to give you all a little bit of a background in how HPE was inspired by this grand challenge and how we were able to take it on as soon as the pandemic hit us. So we've been thinking about drug discovery for a while, right? So we've been thinking about this as a problem that's expensive. Life sciences customers have complained to us all the time that it's taking $1 billion to create a drug. It's time consuming. It takes 10 years. And because of the combination of the two, they can only invest money into research projects that invent new drugs if the market size is more than $1 billion, or if a patent is expiring. They want to come up with some new ideas and so forth. And so they told us about different ways in which they could augment themselves. And some of them was the knowledge of underlying disease mechanism. So ideas about personalizing precise medications to a diverse cohorts of population, having confidence in some of the biomarkers and surrogate endpoints and so forth. And so we've been thinking inside HPE about how do we reimagine drug discovery in general with the computing capabilities, with the exascale era that we talk about and so forth. And we've been trying to figure out what the mechanism was going to be. And then when we started digging into this, we realized this was actually an opportunity for a better healthcare, because the sustainable rates of $1 billion in 10 years is not going to be working well for 6,000 rare diseases that still do not have a cure, that 25 million Americans are suffering. And what was more interesting was that there was this emerging need for attacking infectious diseases that are transferring from animals, the zoonotic diseases. And 75% of emerging infectious diseases are transferring from animals. And interestingly, we just heard about the coronavirus this year, but the fact is we've had 87 new infectious diseases since 1980. That's almost 2 a year. 15 of them have actually ended up becoming outbreaks. And then every outbreak is costing us about $8 billion, and a total of about $8 trillion per pandemic if it's really nasty, like the one we are in right now. And so given the situation, the question then that HPE was posing ourselves was, can we augment what was already happening with clinical trials? which was predominantly an in vitro environment or an in vivo environment where you would do experiments in the wet lab, experiment in a petri dish, take it into animal studies, and then pass it on to humans and so forth. And the process was to take a compound database, try to figure out what the properties of those molecules are in the database, and then go through this process of lead generation to preclinical study to clinical study and so forth. So the idea was, the complex process, several different challenges as we go through this. How can we now take this challenge, augment it with what we call the in-silico approaches of using high-performance computing, AI, and data-augmented environments to take an order or more magnitude bigger set of compounds and go towards safe, efficacious, rapid response, cost-effective, innovative drugs, and in the process, actually shorten that 10-year window. So we've been thinking about this for a couple of years, or maybe more than a couple of years. And interestingly, the way we had started formulating the drug discovery problem for our supercomputing division was that we were thinking about this as a sequence of workflows of search problems. And so if you think about all possible molecules you can make with the elements that you can find on Earth, the chemical space, this is what it's called, is actually 10 to the 60 possible compounds, out of which only about 10 million of them will actually have biological relevance, out of which only 10,000 of them are going to be small molecules that will actually work with the human cell. And then that's actually synthesizable from some natural product or synthesizable from some chemical manufacturing unit and process. And if you take those 10,000, only 3,000 of them are going to be effective towards doing something, and only 800 of them are going to be saved. Right? So that's your needle in the haystack search problem. And when you think about the computational perspective of what it is to start with a chemical space, you can't imagine 10 to the 60, that's way more molecules than the universe. The combinatorial scale of the problem shows up. And then when you start thinking about how many proteins are there on Earth and how do you compare which protein interacts with which other protein and how do you understand the disease characteristics of the host versus the disease characteristics of the organism, that blows up as well. And then for each molecule-molecule interaction, what has happened over billions of years, you're trying to simulate it into a shorter window, and also trying to look at what happens in a nanosecond timescale over a period of time that helps you understand what the chemical process is and how it works and so forth. And while all of these are scientifically inspired ab initio first principle processes, then we've got the augmentation of the expert itself, where you have problem sets like an average hypothesis takes 8 months to formulate. You have to read about 2,500 titles and 250 abstracts and 25 papers before you can come up with one concept that's novel and valid and so forth. And so all of these were adding up, and we thought, what can we do from a computational perspective to address some of these search problems in the drug discovery pipeline? And so when we started digging deeper, we quickly realized that this search was a multimodal, interactive, and an iterative problem. And so the challenge compared to most other business intelligence tools or data science tools was that this was a unique domain that had weird shapes of data. So for example, I want to talk about some of these data sets today. We had to deal with data sets that are coming out of microscopes that model cellular parts of the human body and other organisms. The way that works is you take slices of multiple shots of the data, and then you try to reconstruct 3D from the 2D slices of a microscope. And then you have databases of molecules. Some molecules are SMILE sequences, some molecules are 2D diagrams, some molecules are 3D structures. And then you have scientific documents, which are papers written in natural language. And there's some that's not as structured as a paper, or they're much more, scribbled lab notes and variable measurements taken here and there, structured results from assays and instruments. And then we have the massive genome sequences. And when you think that these are all already different shapes of data, think about now the mix and match of similarity estimation in one, weak correlation with another, and so forth. It's not just the data structures that are different, it's also how you stage this data towards making that leap of faith connection that now you have to start thinking about. So you have to start thinking about data as, here's data that are meant to be files and will remain files forever. You have to start thinking about data that can be curated and posted into databases that you can analyze and have built operational and business intelligence around. And then you'll have data sets that you have to integrate together as knowledge graphs and ontologies that are meant for interactive discoveries, that are meant for scientific process of interacting and creating new insights and creating these leap of faith connections and so forth. And so once you have this data on one side, the process that goes on the other side, the search activities I talked about, is taking a particular molecule and creating different conformers of the same molecule, which could be as simple as doing rotation and translation in 3D space. It could be some kind of folding, which is a shear process that you can impose on the protein molecule. It could be some kind of search for properties like absorption, distribution. And so that decides when you take a pill, is it going to get absorbed in the body? Is it going to be crossing the blood-brain barrier? Is it safe enough that you will excrete it once its purpose is done, and so forth? So you're going to put those molecules into different pipelines that do different things and so forth. And then once you prepare your database, you're going to have to put it through so many different processes. I'm going to talk about them. So I'm not going to dig deep on this particular slide, but you just think of the ability that you should be able to match a SMILE sequence and its equivalent 2D structure. A system that's able to intelligently help you interact with it should understand the same 3D structure in its SMILE sequence, in its 2D format, and in its 3D sequence for a molecule. The same student should also understand the relationship between a drug and a disease, or a drug and a symptom, and so forth. So you need the richness of representation at your data foundation layer to not only stage your multimodal data, but have the ability to stage it in a way where it's much more meaningful when you query it and so forth. So this was the background for what we'd been doing up until this point. And then now we've got the pandemic that hit us in early February, March of this year. And immediately after, HPE leadership announced that HPE will be opening up all its patents to fight COVID-19. And since the acquisition of CRAY had just completed, there was also another press release that talked about how CRAY's acquisition is going to help the COVID-19 research and so forth. So for the rest of the talk, what I'm going to do is talk about 3 or 4 projects that were out of many that happened inside HPE in a detail where you'd appreciate where the supercomputers made a difference and how they contributed towards coming up with some interesting insights. And so the typical problem statement, and again, we collaborated with multiple people, like I said, multiple pharma companies and multiple research organizations that were interested. But the core problem I'm going to walk you all to today is starting with a problem statement that the person that's talking to us has a 200,000 molecule plus database. Some people came to us with 500,000 molecules, but let's say it's big enough that you can do it on your laptop. And then the question is, can you use in-silico methods, just using simulation, just using big data techniques, using AI, pick the 10 most important, useful, ranked molecules that you can take it to the next stage of preclinical/animal testing and so forth. So that is the challenge that we took on. And so before I jump into the details, I'm going to be using some words. So I thought I'll introduce the terminologies first, and so you understand the complexities of the problem. And when I use the terms interchangeably going forward, you understand what I'm trying to do here. So on the left is a picture of the coronavirus. Everybody's probably seen it to death. And then the little red thing that shows up in the inset that I'm showing with my laser pointer over there is actually the protein structure of the spike glycoprotein in the coronavirus. So when I use the word viral target protein, think of that to be some part of the virus that potentially could interact or can be targeted towards binding another small molecule that will then do something to that virus, either kill it or avoid it from replicating, or just make it ineffective and not interact with the human body and so forth. But to get to that protein structure of the spike glyco part of the virus, we actually had to image the virus using CryoEM microscopes. And so that's the structure that's output from processing the virus using a CryoEM microscope with the particular imaging technology that it's either X-ray diffraction or CryoEM, but that's how the data set looks. And then using some other approximation techniques, you get to the protein structure. Then aside from this is the sequence, and this is the protein sequence of the spike glycoprotein, which is much more like a nucleotide sequence of amino acids. When I use the word drug databases, I'm going to be talking about both chemical databases that have structures of known molecules, and then some that are synthesizable but have not been realized yet, that we call virtual molecules, that they have structure, they have a sequence, but nobody has actually made them or put them on shelf. But it's still an idea that, hey, that molecule could be effective as something that will bind on top of the structure. So that's the search process, where when I use the term structural evidence, I want to talk about how once you have the viral protein that's underlying their 3D structure, the small molecule goes and attaches itself to that particular protein to make it ineffective or to make it stop doing something that it's affecting the human body and creating symptoms. So that's one kind of search outcome you expect from from this process. And then the second set of search outcomes is the search for functional evidence. And this is where you're trying to figure out this molecule, has it ever been in the past used or known to interact with some part of an organism or some part of the cell in the human body or in some animal body and so forth. And once you understand that mechanism, you put the different pieces together to explain and justify why this needs to be put on a clinical trial. So once you start searching for structural evidence or you start searching for functional evidence, sometimes you could run into two. So I'm going to talk about some of these workflows that help you take the data, search for structural evidence, and search for functional evidence. And so I kind of divide this into the 4 buckets you see over here. It's a 2x2 setup, if you want to imagine it that way, where typical drug discovery problems fall into this category. And so when I said the convergence of HPC, AI, and big data, and every time I give this talk, folks immediately ask me the question, who's going to be the winner? And I want to kill the joy out of that question right away, saying there is no winner. And the reason is because of the following, right? And so the way these methods are designed are unique to how you're approaching this problem, right? So the first way I describe this picture is that when you think about diseases, you either know a lot about the disease or you know nothing about the disease. And so in the case of COVID-19, I call it an emerging disease target, on which we had very little information compared to what we knew about influenza, for example. And on the other hand, you've got the databases that you're going to be screening virtually to make sure you're able to find a drug in those databases that map to the disease targets, right? And so if your database is well known, as in it contains molecules that have already been put through a trial, they've been, they were efficacious but not safe, they were safe but not efficacious, but you found some other protein interaction mechanism associated with it, or there was an alternative mechanism that actually creates a side effect that was not that was adverse to the problem you were trying to solve. And so when you have these setups and you're starting with a situation like COVID-19, where it's a novel virus, nobody knows much about the structure of the protein or what it does and how it functions into the human body, and you're trying to design a completely new molecule, the need for scientific explainability, the need to explain that from first principles, I can understand why this particular molecule is going to go bind itself to this particular target in the virus is important. Scientists typically tend to believe the simulation approach, to say, yeah, that gives me the first principle design of why it's going to bind. It's either shape, or it's going to be some Newtonian mechanics, or it's going to be some quantum mechanics. They would like to understand the explainability of why the interaction between the drug molecule and the viral target protein is happening. But then the other problem, or the first ray of hope often when you have a disease like that, is to figure out, I already have approved drugs in the market. So we know of remdesivir, we know of hydroxychloroquine, we know of all these stories that we heard in the news, where these are all molecules that were pre-approved for malaria, for example, or HIV, for example. And then you're trying to figure out, hey, I have the similar symptoms as the disease, so can I just repurpose that particular drug for this problem? And there, what you're searching for is some ways to connect the facts, properties about those drug molecules, their interaction mechanisms, their inhibitory behavior, their modulatory behavior, and see how that works with the structures that you've just learned from imaging from a novel emerging virus. And then the bottom left part of this 2x2 matrix is that you know that COVID-19 is like the flu, and therefore you want to design a molecule similar to the one you typically design with the flu. So you're going to assume that this is the closest disease, and therefore incorporate that prior knowledge into designing a new molecule that you can put into the system. So think of this as being the intelligent version of having done the molecular dynamics so many times to create the 800 drugs that are already in the market. So the knowledge that comes from having done the docking on the existing molecules that proved to be safe and efficacious, and the 3,000 molecules that you ended up docking that did not prove to be efficacious. Now, can you use that knowledge to somehow design a mechanism-driven molecule that is close enough that it works for this particular novel virus? And then the last section is much more not as interesting today for COVID-19, but it's interesting from a pharma perspective, is that once somebody comes up with a particular drug, now can you create an alternative with a small edit on it that doesn't have as much of a side effect, or that you're able to increase the dosage and it doesn't harm the human body? Or you're able to create a new molecule for an existing market that you can go into as competition and so forth. So we see that landscape of drug discovery kind of fall into these 4 buckets. And depending on which side of the story the pharma company is in, is if they're in the process of synthesizing new molecules versus reusing existing technology for the new disease, you pick and choose whatever solution you want. And so there's different workflows that different companies work with. So I just captured a few of them here. This is not exhaustive by any means. I'm going to talk about some of these in detail, so I won't spend too much time on the slide. So the first idea is I don't know anything about the disease. I need to at least find out its 3D structure so I can start with first principle simulation calculations. So if somebody says, hey, here is a cell that we believe has the virus in it, let's figure out a way to create a 3D structure that can then be put into these diffraction instruments and CryoEM microscopes to create the 3D structure. And once you have that 3D structure, you want to also prepare your database either from previously published literature or existing databases that different funding agencies put together, and then create the conformers that could potentially be binding with the 3D structure you just estimated. Or once you could take the opposite approach of saying, I'm going to try it against thousands of molecules that I know and see what sticks. That's a computationally challenging problem, but if you have the resources, you can do it as well. And then once you get down to the path, you have to prove that this was actually an active molecule and not a promiscuous one. And so you'll have to prove against decoy molecules of the same structure and so forth, and then actually end up proving that this molecule will, once ingested, exit the body without doing much damage, and so forth. So you've got workflows that work on the same data sets, work on the same multimodal data I talked about a few slides ago, that you want to be able to draw different inferences from the same source. And so when you look at the landscape of tools, this is from a different source called click2drug.org, and they've got a huge compilation of different software tools that one uses for drug discovery. And these are all tools that, I'll be honest, works on a laptop but doesn't scale on the supercomputers, except for the docking ones where the higher the computational power you can feed it, the higher resolution and fidelity you can expect from it. What I'm going to talk about is tools that are not listed on this particular slide for the rest of the talk, and focus on some very unique capabilities that only supercomputing can offer. I'm going to focus on the quadrants again. So I'm going to talk about quickly on CryoEM and how supercomputing can help you with that creation of the 3D structure. I'm going to talk very briefly also about docking, doing first principle simulation. I'm going to talk about building AI models that leverages data from previous docking experiments that actually perform better than the docking experiment itself. And then I'm going to talk about some use cases where we took existing knowledge, both from the data sets that were made available public through the pandemic and databases that have been released prior to the pandemic, to see how those could be repurposed for COVID-19. So I'm going to talk about those 4 use cases for the next 40 minutes or 30 minutes or so. Okay, so the first process is actually creating the digital copy of the virus, which you can start doing your in-silico workflow. We can start imposing your in-silico workflows up. So it starts off with the CryoEM. You prepare your sample, you put it through it, and the CryoEM sample, it's not just like taking a picture. It's like taking multiple pictures, and you do it multiple times to make sure your answers are consistent, your results are consistent, and your 3D structure is exactly what you think it is, because it literally depends. The rest of the processes of drug discovery depend a lot on the 3D structure, so you want a lot of redundancy built into it. And so a typical day produces about 1 to 2 terabytes of data from the microscope. And if you just used a single GPU, it would take you 1 entire week to create one final structure from all the 2D slices to 3D conversion using the Reliant. And so even before the pandemic, HB had announced that we have a reference architecture put together. That's a scale-out architecture that you can pack as many servers as you can, that scales Reliant across the GPUs in a node. And it actually can get you up to 7x speed up if you packed it up enough with high-density GPUs and so forth. So this is a workflow that we accelerated in the past with some of our products, but I'm not going to talk about it today. I'm just going to assume that we've got the image in 3D structures. And so I'm going to focus on 3 different workflows that uniquely use supercomputers now. And all 3 of them assume the 3D structure came out of one of those imaging facilities. And we have identified sequences either from a genomic sequence or from that 3D structure. And those are the inputs for all 3 approaches. So the one on the left is what I call the simulation approach with high-performance computers. This is what high-performance computers were built for: strong scaling codes that grid your 3D molecular structure, that grid your drug molecule, and then impose charges on those gridded parts of the two molecules, and then computes the different kinds of forces, the atomic forces, to figure out will these two molecules come together and actually bind and then interact like they should for biological relevance. So that's the simulation process that uses molecular docking. And the work that I want to talk about uses the source code from Autodoc Vina. And for that code also, we ended up creating multiple ensemble analysis, which means that we not only took for each molecule multiple poses, we took multiple targets targeted by multiple poses, and then computed the ensemble docking capability and binding energies of that entire sample set of conformers from both the ligand side and the protein target side. And then we put it in front of a pharmacologist who would then talk about this docking happened because this particular small active site in the target ended up being the one where the small molecule went and bound itself. So that's the simulation side of the story. The artificial intelligence side of the story is that we looked at a database of molecules that had previously been used for docking, synthetic molecules, synthetically generated docking binding energies, and so forth. And then we asked the question, having done docking so much, having done docking over and over again in supercomputers for the last decade or so, can we now use that knowledge to build an AI surrogate model that helps you predict a yes or no answer to will this molecule bind or not really, really fast? And the reason for doing that is because a single high fidelity resolution, depending on which kind of dynamics you use for your simulation, could take all the way to an exascale supercomputer. And it could, even for that supercomputer, it would take you a week to do 2,000 molecules of screening for a single pose. And so if you really had to do the ensemble analysis and your post analysis, you don't have enough compute in the world to do it. And so we wanted to create a surrogate model that approximates and gives you a yes or no answer, and does it really, really quick, so you're not spending too much time on molecules that are not of interest to you. The third part is where is a unique component where with the simulation part, like I mentioned before, you don't know anything about the target, you don't know anything about the molecule, you're assuming that the physics will dictate the outcome of will they bind or not. With artificial intelligence, we knew a lot about the molecules, we don't know anything about the target. So we're trying to extrapolate that the following features in drug ligands before with similar viruses can now be extended to this virus. So it's you're doing some kind of an equivalence translation using an AI model. The knowledge graph approach is slightly different in the sense that you're trying to put together the different pieces of the puzzle that have already been published with other viruses. So if you want to think about how this one works, we put together capabilities to search through similar proteins of the interest protein that we're looking at with the COVID virus. And that helps you make connections to proteins and other viruses, proteins and subparts of proteins and other viruses, and interactions of those proteins and the other viruses to other organisms and so forth. And once you have this ability to now connect the different pieces, we've put together an integrated knowledge graph that I'm going to talk about in detail of proteins, protein-protein interactions, drug banks, drug molecule synthesis, and so forth, to ask the questions about what properties do I want, what experimental evidence do I have for biological relevance, what safety efficacy data sets can I pull out from this knowledge graph, and so forth. And once you have all these put together, you can say, hey, here are some potential molecules you want to think about because we think this will be a good candidate for the following reason. And so most of the work that I'm going to present was run on one or more of these machines. And so these are the three most used ones. And you could see actually, these are all different architectures already, even though they all say use the word XC. One of them is purely CPU based, the rest of them are GPU based. And so each one had its own purpose. So the first one was a supercomputer served on the cloud. That's a computer that's available in Azure, more typically meant for it's a small scale machine. It's got 2,000 cores. But then it's going to be it's a machine that's meant for simulations in a scale out nature and not the exascale nature. And we ended up running an entire database of about 400,000 molecules on it, and the results were actually published. The goal there was we had the strong scaling code with Vina. We just had to run many of those for different ensembles and different conformers I talked about. And it could actually take weeks to hours per simulation, so depending on the size of the molecule and the size of the ligand. The system in the middle is a GPU-based system used for training our AI workloads, and the one on the far right was meant for running the CRAY graph engine that actually I'm going to talk about later reasoned with 150 billion facts and 30 terabytes of persistent data while in memory. So quickly through the simulation process, and so this is how it works. So with the COVID protein alone, there's 150 plus COVID-19 targets. And these are all different parts, different substructures, different imaging complexes that different researchers have used and made available publicly about the virus, right? So these are all published in a database called PDB. You can go there, search for COVID-19, get one of these structures. And the question then becomes, can you take one of these structures from the database and then look at the database that's also available of potential molecules of infectious diseases, potential molecules that have been previously approved by FDA, potential molecules that have never been tried. So those are three different databases there. So Campbell is representative of molecules that have been approved before. Sweet Lead is focused on infectious diseases, and Coconut is a natural product database, and you can see the different kinds of molecules in it already. So what simulation does is once you assign the different charges, and then once you're able to find where the potential active site could be, you're trying to simulate the dynamics of how two molecules would actually interact in silico, and try to simulate what would happen if they were actually in a Petri dish, for example. And you come up with a mechanism to score that interaction of the charges, interaction of the mass, interaction of the protein from a biological and a chemical perspective. And you could use this core function with different kinds of energy functions. You could use Newtonian, you could use quantum physics and whatnot. And the idea is that the binding energy that you're able to predict from those interactions will help you rank those molecules, and you'll be able to decide on a list of top 500 that you want to go, shortlist and verify. And so we had 87 different projects, and this is one effort where it was not just HPE, it was a consortium from every supercomputing builder and every supercomputing user facility that contributed compute cycles toward the project. 87 different projects trying to come up with different mechanisms to understand how the simulation of the drug-protein interaction problem can be run on different databases, run with different initialization settings, run in different environments, and see which one's the most accurate for predicting the interaction inhibitory activity of the drug. And so we had this blog that came out from UA Edge, and this is Professor Baudry, a friend of mine that I've collaborated over for many, many years. So he was able to take the Coconut database and come up with 125 potential sources. He's actually moved it to a different facility that's now doing the further clinical testing and preclinical testing on that data. And so this was one experiment where somebody that had expertise in the ability to do molecular docking walks in and says, hey, I have these huge databases that's never been tried on any of these sites. A case of an unknown database and an unknown molecule, does it from first principles and identifies the top 100 from a 400,000 molecule database to go prioritize so he can do his research a lot better, a lot more focused in an intelligent way. So now I'm going to move on to the next method, which is the AI-based method. So this is where I'm going to assume that since we know enough about targets, and so we don't know enough about targets, but we know enough about the molecules, can we build an AI model that learns from the previous experiments how good or bad the binding potential is going to be between the 3D structure of a target and the molecule. So if you were to redo all of the experiments of the drugs that succeeded in the market so far with docking, and you're able to learn in the process how that worked, and be able to build an AI model that then tells you, I'm going to learn the structures, the patterns of how hydrogen, oxygen interact, hydrogen, carbon, nitrogen interact. And I want to learn the active sites that are in the target that these molecules are binding to. And without me specifying it, and instead using a graph neural network for it, can I come up with an architecture that helps me do that prediction? So you see the structure over there. That's the neural architecture of PHARML. We actually worked with the Medical University of South Carolina to do this. Dr. Yuri Peterson was the brains behind it. He helped us identify this binding DB dataset, which contains the close to 1 million data points on binding data of 6,200 different protein targets with 378,000 small molecules. And so that was our labeled sample data. And we trained our model on this particular data set with the binding energies as our predictive function. And we came up with a well-trained model that is accurate enough for us to take it to the COVID-19 problem. And so in terms of where we were and competing with the other models, we were one of the early ones that adopted a graph model. We've got a transformer model that actually published yesterday from Georgia Tech. But yeah, so we were one of the first to come up with the graph structure to do it. And when everybody else was still figuring out the errors and generalizability issues with different kinds of validated metrics, we were, because of the nature of how big of a model we could train on a supercomputer, and the number of molecules we could use, the number of proteins we use, the number of molecular protein interactions that we could predict, and the sample data sets we had, we were able to get all the way to a 98.3% accuracy on 25% of a bigger data set that we'd not seen before. And so compared to the other methods, we thought our model was performing better because of the advantage that we had. So you could see we are at least an order of magnitude bigger in terms of the input data, the ground truth data that we used to train to build the AI model to do it. And so when we did it, the first question often is, hey, I've been doing docking for 10 years. You'll have to tell me why I should even listen to this particular model, and should you even believe in an AI model for it? And initially we thought, we got lucky with the structure, and so with binding DB, our accuracy is pretty high, and that, once we figured out the model, we were able to train it in 25 minutes to that particular accuracy, and so forth. And the first question that Dr. Jerome asked us was, how good do you do when I throw in decoys? And so he introduced us to this data set called DUDE. It comes from Stanford. And the idea there was, for every molecule that you have, you also have a similar structure that's a decoy, which is a molecule that will not interact and is known not to interact. And so the question was, how good and how sensitive or not sensitive is the AI model when you do this adversarial testing of the neural network with a decoy data set? And interestingly, the AI model had learned enough that it was doing as good, if not better, as what you would get from a docking approach on a decoy data set. So that gave us confidence that what we had learned was not garbage, and that we could actually apply it to an approved data set. And so we tried taking that data set, trying to figure out how many different molecules that appear in the clinical trials as of June 2020 could have been predicted if the AI model was actually looking at the same drug database and using it for repurposing. And so when we took Kimball, which like I mentioned before, is a database of drug and interactions of drugs with human proteins, we were able to come up with about 15% of the molecules that are in active clinical trials, which is still better than what first principles docking was doing at the time. And we looked at only approved molecules, and we looked at our list versus the list that came out of docking. The number of antiviral drugs we identified was higher. The number of drugs undergoing clinical trials was also higher. And when, for an unknown data set, which we had never seen before, we got some interesting lessons that came out of that experiment. We took about 200,000 molecules of previously never seen before molecules, some virtual, some real, and we tried to come up with which of those molecules could be good binding potentials for COVID-19. And we wanted to find out the top 500, and we wanted to do it really, really fast, right? So we wanted to come up with the answer in less than a day, compared to what it would have taken docking to do over multiple weeks, right? So that was the idea for this particular approach. And the lesson we learned with this particular model was that the model learned a lot about the molecules, but hadn't learned enough about the targets. And so at sometimes, when we introduced it to a newer data set, it was predicting the same properties that you would of a ligand, but the fact that that ligand could potentially be working for this particular new virus was not explainable to us. And so we were trying to figure out how do we improve that process, and we're still in the process of figuring that out. But the good news is, compared to all other AI methods, PHARML was doing better, like I said, in the metrics of number of antivirals in the top list, the number of molecules in clinical trials, and so forth. And these are some papers that I mentioned before on the survey slide, where our AI methods were doing better than the other AI methods published for the deep affinity prediction. And so and interestingly, it's not just the number of molecules. So if you look at where in the ranking something shows up, our method was also ranking the molecules better. And to give ourselves a little bit of an advantage, we were open source very quickly, and we also gave away the data set after having trained it over 64 GPUs for a week. But the development took way longer than that. And the interesting thing about this model is that to work, you actually need to know where your starting point is going to be, where your active site is, and then you try to do your molecular simulation around that active site. That's how much compute you have. You can't do it for the entire molecule. Compared to the docking approaches, our approach with this model, once you train the model and you democratize and distribute it to everybody else, the virtual screening actually takes, you can actually process 9 compounds in 1 second on a GPU and say if it's going to bind or not. Right? And so that's how quick the screening becomes once you do that. And interestingly, from a validation perspective, in addition to the number of antivirals I talked about, the approach was doing better in terms of the probability of binding compared to a random selection of ranked molecules. And we also implicitly learned the Lipinski's rule of 5. A pharmacologist looks at the data and goes, why are you picking molecules less than 500 molecular weight? And like, I don't know, that's what our model learned. And so those are all things that our model had implicitly learned that we didn't tell them to learn. And so this is the side effect of training with ignorance. And so we did get to know that this model had actually learned some of those without us telling it to do so. And so this was released open source, and you could see Dr. Peterson over there. So he's a collaborator that was kind enough to work with us and then make it available for the rest of the community. If you're interested, please feel free to play around with it and give us some feedback on taking it forward. That's the AI piece. So for the next 10 minutes or so, I want to talk about the third leg of how we use supercomputers, which was mostly a data-driven approach to building knowledge graphs. And so very similar problem. We're taking potential drug databases on one side. We've got the drug targets on the other side. The drug targets, instead of taking structures, take protein sequences now. It could be amino acids, it could be the nucleotide sequence, but we take the genomic sequences now and the protein sequences now and try to make the connections to existing bodies of literature in different databases. And so the typical workflow for would then be to say, I'm going to go to uniprot.org, search for this particular protein, find all its interactions, and then take those proteins, go to another data set, and look for protein-protein interactions or protein-drug molecule interactions. Then go to another data set to figure out if it's still safe and efficacious and so forth. So literally, it would have taken a scientist or a researcher multiple queries in multiple databases. And if you wanted to ask the query backwards, it was going to be a cumbersome process. So what we ended up doing was we built an integrated knowledge graph that put together these 13 different databases. And the approximate size of the data was about 30 terabytes on disk, 155 billion medical facts, right? And so once we put this knowledge graph in memory, now we could ask all kinds of questions on it. We conducted multiple hackathons where professors that taught pharmacology, professors that did research in docking would come to us and say, let me play around with this. I have a few questions to pose, and I would like to know, so it's, what's out there in the literature in a few seconds compared to me reading 250 papers over 6 months. And so I'm going to walk you through some of the things that came out of this work. The first one was we leveraged some capabilities from before, and we added new capabilities to it. So we added a component to our existing graph database to handle sequence data. So that becomes a multimodal graph database now that is capable of actually hosting both protein sequences and knowledge graphs, and can now link protein with protein sequence similarity and make those weak correlations of protein similarities to interactions that are in the literature published as a fact in the knowledge graph. And so you're now able to write one query that computes both the protein similarity, does protein sequence analytics, and you can write query using the same language to run a knowledge graph retrieval query. And you can do it both ways. You can start asking different kinds of questions. So the first question during a hackathon that one of our collaborators asked was, so the NCBI has published the genome of a SARS-CoV virus. So what happened? How did this even come to place in nature? And his hypothesis was that not all viruses show up suddenly. There's always something that evolved from, and he wanted to know what part of the evolution from the previous known virus was actually causing the virality this time, and why is it much more dangerous than the previous ones. And so we went back to the data set, and to answer his question of which protein had changed a lot, which protein hadn't changed much, which protein did we have a lot of data that we knew from the previous viruses that we have molecules that can go attack it. That was the first line of thought to figure out what is my ideal target, where should I spend my computational resource on. So we have that result at the bottom where you have the NSP set of proteins that are on the side of the sequence in the genomic browser over here. And then we've had the envelope, and N proteins, S proteins, and so forth. And so we were able to figure out that there is, significant evolution on the S protein and the SB3, the two main proteases that most research has been happening around. And the virality is probably caused by those two. And so we knew why those two were good targets for us to test all the known drugs against. So once we knew this answer, literally we could come up with this answer in less than 30 seconds. And the fact that you could search not only a Wuhan against the SARS-CoV-1, we could now, because of the parallel nature of the supercomputing problem and the database that was hosting it, we could do the exact 4 million all-to-all comparisons of protein sequences to all known protein sequences in the world and come up with an answer every query. Right? So think about writing a query, giving a protein sequence as input, and that comes back with results of having compared against 4 million other sequences, subsequences of virus, subproteins of viruses that you've known before, subproteins of bacteria that you've known before, and so forth. So that's the case we had now that we didn't before the pandemic. And interestingly, the first question as a scientist is this even valuable for what we are trying to do? And so the question was, would you be able to find out, potential vaccines and potential drugs just by using this particular tool? And interestingly enough, we were able to. And so when one of our hackathon collaborators said, hey, the first ever vaccine for smallpox, actually the first ever vaccine, was discovered because people found that folks that were dealing with cows and cows with cowpox ended up not getting smallpox. And therefore, small dosages to the cowpox virus would actually create immunity for smallpox. And so the question was, will our system be able to extrapolate something like that to create a hypothesis? And not only did it do cowpox and smallpox, we could repeat the exact same thing with Zika dengue. We could also do it for chickenpox and shingles. And this kind of gave us the confidence that, by just adding the capability to do protein sequence analytics to an existing graph database, we are now able to make connections across diseases, across viruses, and we can avoid the silo problem of, I'm a researcher that knows only viruses, and I don't know enough about bacteria, so I want to ignore everything that people have published about bacteria that doesn't apply to this virus. So we were able to break some silos there by broadening our scope of search. So that was one. And then we took it up a notch even higher. And this is where, when you think about the first step we did, where we linked proteins of the coronavirus substructures, proteins of, we were able to come up with which proteins map to what other proteins that are known. So that's the list you see over here. And we used this Smith-Waterman algorithm to create that the weak hypothesis, weak inference link to map a protein-protein similarity function. Once we did that, we were able to find out what activities in a host organism were these proteins influencing, or what were they interacting with the host organism, and we could come up with a few. I'm going to talk about tetanus toxin in a minute, but you could see the ACE2 inhibitor that the first initial research on the spike protein started talking about with hypertension medicines and so forth, and you could start seeing the different activities. And so if you actually looked at and researched all the proteins on the far left over here, these are all proteins of the previous virus, the bat coronavirus, the MERS virus from the previous version of SARS and so forth. And so we were able to, logically and explainably figure out that there are protein structures of this coronavirus that's common to other protein structures, and that protein structures were involved in different organisms. And once we were able to bring in UniProt data with the Reactome data, with the drug bank data and the Chembl data, we could hypothesize what molecules in those databases have known or published activity that now will inhibit this activity of the protein-protein interaction with the disease-causing organism. So now by connecting the dots that walk across databases, you could ask the question both ways. You could say, I've got this part of substructure in the virus, find me all drugs. Or you could say, I have this drug, which part of the substructure of the virus, will it be effective against, or will it even work against, or will it bind against? And so we were able to come up with a list. And so the numbers that you see on the far right, and these are the 66 molecules that we came out as a hypothesis. The ones with numbers, I believe, should be about 20. And then the number within the curly braces are the number of clinical trials across the world on that particular molecule, right? So that already tells you that we're doing pretty well in terms of finding out repurposable drugs using this knowledge graph approach. And so I'm just going to take a couple more minutes to finish this. And so this is one step where you were able to connect the dots of evidence based on curated knowledge bases that came from subject matter experts. But then once the pandemic situation happened, a lot of preprints were coming out, a lot of other papers were coming out, and the White House actually released a corpus of documents to the community to be able to use them for other purposes. So Paulo that introduced me, his team did some really interesting work in building a Q&A system around that data set. And folks are welcome to try that and contact Samindu if you guys have some questions around the system and how it's built and so forth. So the idea there was once you're able to ingest a corpus of documents, now can you add a natural language processing inference tool on top of it to ask natural language questions that gives you the search for functional evidence I talked about before. So if I say that tazobactam is going to be treating the coronavirus infection, can you come up with different explanatory pathways that are there spread across multiple papers that tells you what the potential mechanism could be? And when a subject matter looks at it, expert looks at it, he could pick and choose which ones are explainable and which ones aren't. So we could put together that storyline as well. So in summary, I think what I want to tell you is that we were able to take this knowledge graph approach, take the NLP approach, and give the subject matter expert what takes 2,500 papers to happen in seconds. And as of June 20th, we were doing about 30% success in hypothesizing a particular molecule that actually ended up going through a clinical trial. And we also have an interesting hypothesis that has strong correlations that the tetanus toxoid vaccine is actually the one that's mapped to reduce severity in COVID-19. And there's been some research on BCG, but there's not enough on COVID. And we're actually seeing that when countries support a tetanus booster, their asymptomatic rates are a lot lesser. So in the sense that if you are vaccinated and your vaccination is current with your booster, you are more asymptomatic than you're not. And for folks that are suffering from severe long-term effects of COVID-19, there are similar symptoms as tetanus would have caused otherwise, neurologically speaking. There are some interesting correlations that are emerging from this research that popped out of a hackathon exercise with folks. And so I'm going to stop on this slide that I talked about 4 use cases. There's a couple more that I couldn't mention, but these are the efforts that happen inside HPE, where we were able to use supercomputing. I gave access to supercomputers to folks that knew how to run molecular dynamics for completely innovative first principle thinking. We were able to build AI models that replaced folks that couldn't do the docking approach. We came up with ideas to create new hypotheses with natural language processing tools and knowledge graphs. And we also have a couple of other workflows and reference architectures that help you learn in a distributed way when you couldn't move data, but you had data streaming in at different hospitals. The swarm learning approach over here actually does that, and there's a paper that's coming out in Nature pretty soon that talks about that particular approach. So with that, I'm just going to stop and open up for questions. And to you, Jay.
Hey, Rangan, there was a great talk, very, very interesting, and opening a lot of interesting opportunities to accelerate fight to the coronavirus. There's we do have 5 or 6 questions on the chat. I'm going to start going in order, roughly speaking, and then we'll see if we want to come in. The first one is. There's a lot of noise in the background. I don't know if it's me or if you have a speakerphone on, by the way. Okay, so first question is it clear in the beginning of the work that we're looking for a drug that eradicates a disease, or one where we make an annual vaccine like the flu?
So we had collaborators that were looking for both, right? So the vaccine people were doing vaccine work. There were also researchers that were looking for combinational drugs, not just one. So this is where it depends on the type of question you are starting off with, right? So if you're starting off with vaccine, you had a slightly different approach to doing the workflow. If you're starting off with combinational drugs, you had a slightly different workflow. So I couldn't capture all of this in the talk today, but the early approach initially with HPE was we were open to any idea of anybody that was walking in and saying, we would like the following resource to do the following workflows. And so most of those vaccine research work did happen as part of the consortium. And I wasn't personally involved in them, but I know that the vaccine research was happening there.
All right, let's go to a different question. One about the execution mechanism. You talk about running most of these things on supercomputers, but the question that probably a lot of other people have is, do you actually need a supercomputer, or are these problems massively parallel and loosely coupled enough so that they could be distributed to like a cloud infrastructure at the internet scale, which, you know, in aggregate probably has even more compute resources, or do you actually need what a supercomputer brings to the table here?
Yeah, that's a great question. So I talked about three workflows, right? The first one was simulation, the second one was AI, and the third one was knowledge graphs. So I'm going to give you the caveats of when you need a supercomputer and when you don't. So the first one, the docking approach, we had simulations being run on the world's fastest computers like Summit at Oak Ridge, and we also had ones that are being run on scale-out clusters. I think for problems where you are trying to come up with extremely high fidelity resolution, extremely high reproducible physics that you were trying to represent in the molecular dynamics. So those were the ones that required your supercomputers. And once you got to understanding that, yes, this is a potential molecule, when you were trying to do the ensemble analysis, and you're trying to do the post identifications, and you're trying to do the active site based confirmatory analysis and so forth, you don't need the supercomputer in the supercomputer sense. You could get away with doing approach or scale out approach in a smaller cluster and so forth. So there, I think the need for fidelity and resolution is the one that went into the supercomputing need. With the AI model, and this is where I think I thought I did emphasize, but maybe I missed the point. The point was that the AI model's training required the supercomputer. We needed, like I said, 64 GPUs running for multiple weeks to train that model. And once we trained that model, now the model is available, right? So anytime we retrain it for doing something else, the transfer learning just. So the expertise to create the model took a while, but now that we've created the architecture and now that we've made it available and we know that people are using it and making modifications to the architecture to do other things, now it's a lot less compute. And that happens in a small scale DGX box or a single node of GPUs and so forth. And you don't need the scale out cluster there. For the knowledge graph effort, can this be done without a supercomputer? Absolutely. It won't be interactive, right? So when I talked about the graph database, it's not just it's a query system, it's also a whole graph analysis system. So literally you could do graph analytic algorithms and pattern search algorithms on the same graph database. And then you can do protein sequence analytics too now that you have that capability. So to be able to do the protein sequence analytics was happening embarrassingly parallel. But then once you made that link for every query, the whole graph algorithms kicked in to be able to connect the dots for you and reason like it was natural language, right? So for those parts, if you wanted interactivity, if you wanted that my discovery is not going to be siloed and I don't have to run 10 parallel queries to come up with a connected insight path, I think you're better off running on a supercomputer than on the cloud. But if you're okay with waiting for maybe 10 times longer, because that's what you can afford, those workloads now do work on the cloud. And so the example, we did port it to make it work on commodity hardware. So we know that it's available for folks if they want to use it again. And this was just more to reduce the time that we were running against for this particular pandemic to come up with solutions fast, rather than, can we do this in the cloud? So we know that now each of these codes can be ported into more effective architectures. But the first try was on a supercomputer. So I don't know if that answered the question. It's a long-winded answer, but that's how I would tackle it.
I hope it does. So since you're talking about AI, is another question that is, can you comment on some other current or possibly future applications of AI in medicine, healthcare, life sciences in general, the one you consider most promising? It's an open question, but just a few ideas that you may have.
Ooh, that's a great idea. So I think the two top applications that come to mind, and one I didn't talk today, was the idea of molecular synthesis itself, right? And so there, I talked about the 10 to the 60 possible combinations. And so I think the future of potential drug design is going to depend on smart ways to generate new molecules that you didn't know you could create and you didn't know could actually solve a disease, right? And it's humanly impossible to come up with those patterns. So that's in the drug discovery pipeline. So if you took a step outside and said, I'm going to look at healthcare and life sciences in general, I think the AI methods, if I were to build one AI model, that will be around how do I deliver medicines in a precision prescribed way, So if I knew somebody's genome, if I knew somebody's Apple Watch statistics, and if I knew somebody's previous history from their medical records and so forth, can I now predict the dosage, the drug, and the molecule uniquely for that person, and can I manufacture that? So that would be the ultimate goal, right? And if you flip the same problem around, and this is kind of my pet project, I think we now have models for the average. We don't have models for the individuals. And I think the future is going to be each one has an AI model, a proxy of himself that represents that person in the healthcare system. So we'll see how that happens, but that's a little bit of an imaginative thought.
Let's see, a couple of more maybe point question. One is when you're talking about the interactive AI-based analysis, there was a lot of yes and no answers. Are you recording them so that you can use them in the future and not be asked again?
This is an excellent point, right? This is where we allow the end user to put in their inferencing tags. So if they said, I don't like this particular answer because that because is there, we try to help them word that in an inferencing rule that goes back into the system as knowledge, right? So we've done it manually right now. So we work with during the hackathon when somebody says that, we try to do this, but we should be hitting attention to how to automate that process of a negative label that somebody gives us when they discard a result. That's a great question.
Then I have two questions on the binding problem. I'm going to ask the easy one first, and that probably is a simple one. Is the binding problem solved using quantum mechanics or something like density functional theory? So in other words, what is the foundation of the algorithm of the binding problem?
So the one that Jerome was using, Dr. Baudry was using, was density functional theory. The one that was run on Gen C was using a quantum approximation to the problem. So yeah, so that was also run on a HP supercomputer, but I didn't talk about it today. I just hand waved and said there are folks that are doing it.
Then there's a deeper question on the protein binding prediction using machine learning. Was there any work to investigate the explainability of the prediction? This is a pretty hot topic in AI these days. And this is, to help, this could help understand what the models have learned for validation, refinement purposes, and in general, whether you can trust AI or not. So I guess it's the explainability and interpretability question of the prediction of the model.
So we are still in the process of doing it, but I'll give you two anecdotes that happened. Right, the first one that happened was so we did find a molecule that was used for a completely different purpose in our list. And not only did we find that one molecule, we also found several variants of the same molecule and ranked them higher. And so the chemist that was looking at it came back to us and said, why did you do that? Because this molecule, when it tried docking, didn't work. And so when he went in, took that particular molecule that was so high up in the ranking and tried to do all possible poses of docking, he came back and said, you know what? There are 5 different active sites on the protein. And we were able to make sure that we were able to confirm now that if you had chosen a different active site, this molecule was going to go bind there, right? So without our knowledge, and we knew that we were active site agnostic by design, but we didn't know that we were learning patterns and substructures of the hydrogen, oxygen, nitrogen, carbon bonds that actually map to different possible active sites. So we had somehow reduced the combinatorial problem of shape into a pattern that we don't understand yet, but we believe it's being captured in the model. So that was one anecdote that came out for the explainability approach. And the second one was we did take the output of the model and figure out how close and how far are we from known prior docking binding energy. So if we predicted the same things, and how good are we with the correlations and so forth. And the results were mixed, to be honest with you guys. And so we're figuring out why and so forth. And so those are things that we're still investigating.
All right, I think I have one last question, unless something else pops up, and that was from me, and that is still about AI. And talked a lot about the training phase and how long it takes. And as you know, there's a lot of discussion about AI specialized accelerators, but their target are usually sort of internet, consumer images and videos. So the data types are quite different that they target. Do you see that they can play a role here to further accelerate the discovery process, or do you have any suggestion for the people building these accelerators, or how to make them more viable for these domains that are a little different than what they're usually targeting?
Absolutely. And so, great question, Paul. So even with our graph model, PHARML.BIND, so it's a graph neural network for the first time, right? And we were trying to see, and this is part of a different experiment we're doing, trying to figure out, based on some of the in-house accelerators we have access to, what does it take to up that number I told you, where with PHARML.BIND in inferencing mode, we could do 9 compounds a second. Now, can we beat that number that happens in a GPU with some of these accelerators? And interestingly enough, we realized that we had some custom kernels built in, because we were not using the traditional sigmoid that comes packaged with the Keras that particular vendor supports. There's issues like that, and then there were issues like once we did try doing it, there were assumptions that these accelerators make on the quantization, assumptions that these accelerators make on the type of pruning algorithm, the sparsities that they can leverage from the model and so forth, that kind of throws off a little bit from the accuracies you get if you do it on the same GPU that you're trained on. So interestingly, if I were to give them some advice back, it would be to add some support for 2D, 3D structures as input, to add support for the ability to have these kernels that do the approximation, and not assume sparsity and quantization for 3D structures where you need double precision. So those will be some pointers that I would point to.
Excellent. I don't see any more questions. So Dan, I don't know if you want to take it for some closing comments.
Yes, I would. Thank you very much, Rangan, for excellent presentation. I really enjoyed it. Thank you, Paulo, for asking the questions. And thanks, audience, for joining us. This talk will be, it is recorded, and it will be produced and then made available at our playlist on YouTube. I would also like to announce that according to our plan, we have a very related second talk, which will take place sometime in November. Jelena Pješevac-Grbović from Google will be talking about software related to pandemics. So please stay with us, and looking forward to the next talk. Have a great evening, and thanks again for joining.